Imagine a patient in the intensive unit. Their vital signs are crashing, and the bedside alarms are blaring. This patient is entirely dependent on total parenteral nutrition. An unfortified intravenous IV line is the only thing keeping them alive. Without warning, their liver begins failing, rapidly filling up with trapped fat. It looks exactly like a severe acute case of non-alcoholic fatty liver disease. At the exact same time, their skeletal muscles are disintegrating. The muscle tissue is breaking down and leaking massive amounts of the enzyme creatine kinase into their bloodstream. To the medical team, this combined liver and muscle collapse looks identical to a severe toxic reaction or a rampant fulminant viral hepatitis. They scramble to order huge toxicology panels and viral screens, looking for an external invader. But there is no virus. There is no hidden poison in the IV line. The medical team is blind to the fact that starvation is destroying this person's organs. Searching for a nutritional deficit, blood is drawn from the patient's arm to test for a shortage of the building block choline. The results return perfectly normal. The lab machine reports a healthy concentration, prompting the doctors to confidently cross choline deficiency off their list. They are wrong. Despite the lab report, the patient is actively starving to death at the tissue level for a single molecular building block. To understand why the liver failed, the muscles melted, and the machine lied, we have to deconstruct the architectural purpose of the missing molecule. Relying on a laboratory printout without understanding the underlying tissue architecture and pre-analytical chemistry can mask a lethal starvation state from even the most astute clinicians. Choline operates as a permanent quaternary ammonium cation. This means it carries a fixed positive charge that never turns off. Because of this chemistry, it cannot slip through the fat layers of our cells. It is trapped wherever it is, unless moved through a highly specific transporter door. 85% of the body's choline acts as structural bricks in cell membranes. The body uses a high-energy spark to force this charged molecule onto a lipid tail, creating phosphatidylcholine. Its cylindrical shape allows millions to pack side by side, forming stable, non-leaky bilayers. This specific geometry is the structural foundation required to enclose complex internal organs. Without it, human cell walls would simply dissolve or crack. The liver relies on these bricks to act as a metabolic factory. It manufactures fat, but it must package that fat into cargo ships called very low-density lipoproteins to export it into the bloodstream. Because blood is mostly water, these ships require a continuous outer shield of phosphatidylcholine to navigate without coagulating. Other systems aggressively siphon this material. The lungs use it to manufacture surfactant to prevent their air sacs from collapsing, and the brain converts it into acetylcholine to drive motor reflexes. The most extreme siphon occurs during the third trimester. A fetus will pump choline against a steep concentration gradient, draining the mother's reserves to physically construct its expanding neural network. To survive dietary shortages, the liver has a backup generator called the PEMT pathway, which can manufacture the molecule from scratch. However, this pathway is gated by estrogen. Men and postmenopausal women lack this high estrogen buffer, making their liver function entirely dependent on daily dietary intake. This returns us to the TPN patient. Lacking dietary input and a hormonal failsafe, their liver cannot build the VLDL holes. The fat is permanently trapped, pooling until the liver tissue suffocates. If the patient's tissues are starving for this structural brick, we are left with the central paradox. Why did the blood sample show a normal choline level? The answer lies in where the choline actually lives. Over 90% of the choline in your blood is integrated into the physical cell walls of the blood cells themselves. In many hospitals, a blood sample is drawn and left unstabilized at warm room temperature before it ever reaches a machine. This triggers ex vivo phospholipase autolysis. In a warm tube, active enzymes keep functioning, breaking down the cell walls within the sample itself. As the blood cell walls literally melt inside the test tube, they leak their stored choline into the surrounding fluid, artificially inflating the concentration. When the machine tests this melted fluid, it generates a false normal. It blinds the physicians to the reality that inside the patient's body, the structural walls are collapsing. Beating this autolysis trap requires a strict protocol. The blood must be drawn after a 12-hour fast directly into pre-chilled EDTA tubes. The tube must be buried in crushed ice immediately at the bedside to completely freeze the internal enzyme kinetics. The lab must centrifuge the sample cold within 30 minutes and instantly freeze the separated plasma at -70 degrees. Only then can it be accurately measured. Liquid chromatography tandem mass spectrometry is the gold standard, counting the molecules in a perfectly preserved sample. With a preserved sample, the diagnosis is clear. A fasting-free plasma reading below 5 micromoles per liter, paired with a massive creatine kinase spike, confirms a structural breakdown. Accuracy in clinical pathology is secured at the bedside, not the lab bench. It requires manipulating thermodynamics to effectively stop time before the sample ever reaches the machine. Once identified, the instinct is to overcorrect by swallowing massive 10-gram oral megadoses of synthetic choline. But the gut transporters had a strict capacity limit. This bottleneck forces the unabsorbed excess to pool in the intestines. Anaerobic gut bacteria feast on this pool, converting the choline into a highly volatile gas called trimethylamine. This triggers a metabolic panic, resulting in crashing blood pressure, abdominal cramping, and a pungent fish-like body odor clearing through the skin. To save the dying TPN patient, doctors bypass the gut entirely. They administer intravenous acetylcholine or alpha-GPC, which plugs directly into the internal cellular machinery. The resolution is rapid. The liver resumes building VLDL hulls, exporting the trapped fat and restabilizing the melting skeletal muscles. Choline provides the physical architecture for human life, enabling everything from lung expansion to liver fat export. This case proves that fixing a crumbling physiological building requires understanding the chemistry of the brick.